Hello, everyone. Welcome to today's live broadcast, Bioburden Testing at Various Stages of a Biomanufacturing Process. I'm Shannon Stoltz, Senior Editor of Special Projects for LCGC, and I'll be your moderator for today's event. We are pleased to bring this webcast presented by LCGC and sponsored by Cytiva. Cytiva is a global provider of technologies and services that advance and accelerate the development, manufacture, and delivery of therapeutics. Cytiva brings together a wealth of technical expertise and talent, a broad and deep portfolio, and exceptional service to help researchers and biopharma advance therapeutics at every stage from discovery to delivery. We do have a few important announcements before we begin. First, this webcast is designed to be interactive, so we encourage you to ask questions throughout the event. You can submit your questions by typing them in the Q&A box, which can be found at the bottom of the video player. You can enlarge the slide window by clicking on the small icon in the bottom right corner of the media player, and the slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark help widget in the top right of your presentation window. I would now like to introduce today's speakers. We are very pleased to be joined today by Patty Lee and Eric Bjornold. Patty has over 25 years of experience in sterile filtration and quality control. Her education is in microbiology, and she has started her career as a QC microbiologist. Currently, she is Global Product Manager for Lab Filtration Microbiology. And Eric is Principal Scientist at Cytiva with a background in spectroscopy, imaging, and electrophoresis. Eric received his PhD in materials science from Chalmers University of Technology, and he has over 20 years of experience in this industry within different roles. Thank you both so much for joining us today, and please get us started.
So there we are. So I'd like to thank you for joining us. I'm going to talk a little bit about microbiology, where it fits in the manufacturing process. And then I'm going to delve into some of the developments, kind of a product portfolio development, how it changed over the years to actually meet the needs for pharmaceutical testing and other microbiological applications. And then I'm going to hand over the presentation to Eric Bjornold to talk about actually his advancements for reading the colony growth results. So we're going to cover microbiology testing. We're going to look at where the contamination risks are in the system. And then we're going to talk about innovation. So look at how a portfolio has developed over the years to help mitigate the contamination risks and provide the most accurate outcome. And then we'll hand it off to Eric for discussing about how his advancements can help streamline plate counting. So when we talk about microbiology testing, the most important reason that we're all doing that is to protect the products that we're putting out to the field, whether it's the production of pharmaceuticals, looking for spoilage in food, or organisms that could affect the taste of beverages, and of course, anything that might cause illness once our products are released, whether it's water, pharmaceutical, or food. We try to avoid any of those incidents because you don't want to have loss of reputation to your company. You don't want to impact supply chain. You don't want batches of pharmaceuticals going on hold, which can be a very costly event. And you don't want it to result in loss of business or loss of business to competitors, as well as potential fines and penalties depending on the situation. So accurate and reliable detection is important to those individuals that are actually working in the microbiology laboratory. When it comes to the types of samples that are tested during the manufacturing process, of course, the raw materials and the water that go into the manufacturing process, the environmental conditions around the actual manufacturing process, that includes the air and air handling systems, surfaces that the products could contact, or surfaces that actually the manufacturing personnel contact, and of course the individuals themselves that are manufacturing the products. One of the most important points is looking at the purified water system and ensuring that a biofilm hasn't developed in the system. And then, of course, there are special projects for the development and formulation of the product. And during the processing, the intermediate samples may be taken just to ensure that bioburden is being managed during the manufacturing process, that any steps or filtration going on during the middle of the manufacturing process are in fact controlling the bioburden load. And then of course there's finished product testing. So looking for, depending on the type of preparation that's being made, the bioburden resident in the final product. Could be absence of specified organisms, depending on how the product is used. And of course, for those products that are injected or infused, then you need to have absolute sterility of those products. When it comes to the application that we can that we're going to discuss today would be membrane filtration technique. And it's a product, it's an application that's actually stated in a lot of the pharmacopoeias as a preferred method for actually sample preparation. But it has its origins in the water testing realm, where it was first employed for managing public health and safety of the water systems. So it's a popular technique for actual sample preparation. Anything that is aqueous in nature or can be dissolved completely in an aqueous buffer can be analyzed by membrane filtration technique. You put your liquid sample into the funnel, you filter it through, you rinse down the sidewalls of your filter funnel, and then you access your membrane for plating, and then ultimately you grow the colonies. The advantages for membrane filtration technique include being able to process a larger sample volume. The traditional methods that involve direct inoculation, you're limited to the sample size, but with a membrane filtration technique, you can process a larger sample, which might be important if you're expecting a low resident bioburden in that sample. Then the organisms that are captured on the surface of the membrane can be rinsed down with various buffers, making it a very versatile type method. And any of those rinse buffers can help with neutralizing inhibitors like preservatives or antimicrobials. And just to restate that it is it's a very versatile method, because you have your selection of different types of membrane filters to choose from, different pore sizes, as long as they're 0.45 or smaller for pharmaceutical. But when you get into food and beverage testing, you may want a 0.8 micron filter. And then, of course, typical rinse buffers are phosphate buffered saline or normal saline. But when you are working with pharmaceutical preparations, the pharmacopoeias list other rinse buffers that have ingredients to, again, neutralize inhibiting substances. So it's a very versatile type application. Now, looking at where contamination risks reside in the whole process would be, one of the first places is water systems. Resident bioburden that could build up within the purified water system is definitely a risk, because that water is used as an ingredient, an excipient ingredient in your preparation or for cleaning procedures. And of course, the environment that's surrounding the manufacturing process, that would be the air. Is there any organisms that are coming from the air handling system or shutting off of the individuals and becoming airborne and settling on surfaces that that they shouldn't. So surface contact plates, those may be agar plates that are being used. Of course, the cleanliness of the containers that are being used to maybe handle bulk ingredients and or filling into preparations. And then, of course, the manufacturing equipment itself. So the microbiology process will look at the raw materials that are coming into the system, as I mentioned before. You look at the intermediate products, so at certain points during the processing, samples may be taken to make sure that bioburden is being controlled. And it could also be other fluids that are introduced into the system. And then, of course, finished good products. And then one of the most important risks to look at would be the any people that are involved during the sampling, handling, filtration, so the laboratory personnel, and then the SOPs that are being followed during the testing of all of the materials from the water system, environment, or materials. So with membrane filtration technique, we look at, what's happening at the bench level. So they're following regulations and protocols. They have validated methods for handling the samples. The laboratory space may be a concern, the number of people that are in the lab and sharing equipment, the number of tests being processed through the laboratory, and any budgetary constraints or laboratory waste. As a company, when we're looking at that as a whole, we start to look, how does this flow into the development process for products that we could help with streamlining this whole workflow process. So we start to look at, at the bench level, depending on the different industries, how are they processing their samples? Are they using reusable hardware? And does their workflow look different than someone who maybe needs a partial disposable or a completely individual use, ready to use disposable filter funnel? So when we started to develop our product called the MicroFunnel filter funnel, it started to segment into 3 different groups. We started over on the left with a standard filter funnel, and over time listening to and observing customers' workflow and feedback on needs or unmet needs, it started to evolve as a product portfolio. So where does it fit when we look at it a little more graphically? If we look at the top bar, we see that filter funnels are used widely for any sample that arrives at a laboratory that's liquid or can be thoroughly dissolved in a buffer. But then we started to get feedback on from laboratories that needed better handling of the product. So more bulk, being able to transfer items into a clean room, a hood, or a testing isolator. So we started to develop a packaging variation of the standard MicroFunnel. And then we started to get feedback that, hey, we need a product that we can better sample our water system, since that's generating many samples, on as many as 3 shifts during the manufacturing workday. So when we started to think about how do we better design a disposable filter funnel, we had started out with a typical design where you pried the product apart, but we started to look at this and say, what are all the unmet needs? So we designed a product called MicroFunnel filter funnel. And some of the details that, may go unnoticed a bit would be, where are the graduations marked on the cylinder? Well, they should be marked on the outside of the cylinder, and for the MicroFunnel they are, because you want a nice smooth surface for rinsing. You don't want to have any of that thought that, maybe your sample or bioburden got held up on a textured surface inside the funnel. The next thing we thought about is, how do we make it easy to remove the filters? So there's definite design benefits that help with placing your forceps into the funnel and actually grabbing the membrane filter for plating. And then we thought about, how do we process this? We know they have to put it onto a manifold. So the original design was made to go directly on a tulip style manifold. We'll see an example of that in a couple slides, and I'll point that out to you. But then we started to think, well, gosh, all the supporting materials that we put around this, maybe we should provide a better, more secure fit to the manifold. So we'll also see that later, how this whole portfolio kind of progressed to help the microbiologist process these samples in a secure, contamination-free way. And we, of course, offer it in both sizes, 100 ml and 300 ml. So we see here one of the other features, but you kind of have to see it in a visual format. So the MicroFunnel was specifically designed to be ergonomic, make it easier for the analyst to process hundreds of samples in the daytime and not get a repetitive motion injury. So if you see the funnel on the left, it has its lid in place. It holds the geometry of that cylinder. But once you remove that lid, it's a simple squeeze to access the membrane. By squeezing that, it releases the cylinder from the base, and you can simply tip it away and access your membrane. We see the microfunnel securely installed on the manifold. We'll talk about that later, as well as the Centino pump that's on the right. And we'll talk about that also, how those kind of evolved from a workflow standpoint. So again, we see the easy access of the membrane on the left. And then we also have designed in for this product, but it's usually not used for the pharmaceutical industry. Usually in pharmaceutical, they remove the membrane and plate that on an auger. But in the beverage industry, they may actually load the media through the top of the membrane and displace the water in the pad with the membrane, and then collapse it down to a Petri dish. All of these ideas were to, encompass what the various microbiology labs are doing in their varied workflows. So the next evolution of MicroFunnel came with the packaging, which we call MicroFunnel ST. I like to think of that as sanitary transfer. It's much easier to be able to wipe down one overpack bag with multiple individually bagged products inside than it is to individually wipe down each product. And this was highlighted as important for handling materials going into clean room laboratories, hoods, and even sterilization isolators. And then the final evolution of MicroFunnel occurred with the MicroFunnel Plus, where individuals were expressing that they needed a product that could actually collect the sample as well as filter the sample. So MicroFunnel Plus came to be, and it's the only product on the market that can act as both the filter funnel and the sample cup all in one product. It has a vented lid that once you collect the sample, you snap it down to a liquid tight seal, and you don't have to remove it to complete the filtration, because the vented lid allows air to displace the fluid that's being drawn out when you take it back to the laboratory and put it on the manifold. You also see the underside of the funnel. You see that little vent tab that is also protecting the base from any contamination while you're carrying your sample through the plant and back to the testing lab. It reduces waste because it is your sample cup and your filter funnel. And once you get back to the laboratory, you remove that tab, you place it onto the manifold, and you can do your filtration. You don't have that extra pour and wait step, and that pour step could be a potential introduction for contamination that you don't want to accidentally put into your process. The other important thing for MicroFunnel Plus is the vented lid and the vented base allow you to actually sample from hot water loops up to 90 degrees C. So that was an important development of the product, just to streamline and make this product more useful for the microbiology laboratory. Another slight evolution of the MicroFunnel Plus was adding a sample port in the top. And through that, you could actually attach a sample tube to that port and to a micro barb on the various points of your water system for sampling. That further protects the sample while it's being collected, and just a further development for that portfolio to make it more useful for water system testing. So that's the evolution of MicroFunnels, largely driven by feedback from customers, going out and observing the process for microbiology and how they're actually handling the products. Another key development was the microbiology manifold. So in the pharmaceutical industry, it's very common to clean and sterilize, autoclave your manifold system. So we wanted to create a manifold that could facilitate that for a thorough disassembly, cleaning, and autoclaving. And in fact, this manifold comes apart with no tools. You can take it apart in probably a minute or two, because all of those fittings slide off. The way that they're connected together has all of the O-rings on external surfaces, so not in the fluid path. And all of the areas where the fluid does contact are polished surfaces, so you don't have to worry about a textured surface that Bioburden can start to easily build up on. So it's a really nice product, a very modular design. You have your selection of different connectors depending on the type of filter funnel that you're using. We saw it earlier with the MicroFunnel adapter, which we also see here. And this also highlights a coupler. So you can easily extend your manifold to be a 3, 6, or a 9-place manifold. And in fact, you can operate a 9-place manifold without causing degradation of the vacuum pull at the port that's farthest from the vacuum source. And if you can follow up with us later if you'd like a technical article sent to you about our study on that. This just gives you another view of the adaptability that we have for different filter funnel use. And then there's the Centino microbiology pump, which was another idea to make it easier to set up for filtrations. It's used in busy laboratories that may have limited space. It works on main power or battery operated, but it has a disposable fluid path. So when you're ready to do your filtration, say you're in a busy lab and you have limited access to the hood and you get the call, hey, we're finished with our QC testing. You can get in here with your special study. You can just wipe that down, transfer it right into the hood, and be ready to go with filtration. It uses peristaltic drive to pull the fluid in one direction and then right to drain, so you don't have to worry about any pressure backflow. And it just is another addition, another progression of the product, trying to streamline things and make it simpler for the microbiology lab. So at this point, I'm going to hand over to Eric, because everything I've been talking about has been the sample prep step, but the ultimate goal is to actually create growth on a membrane product. And Eric's going to talk to you about how his portfolio can help with reading.
Thank you. Thank you, Patty. I think you can hear me okay. So hi everyone, my name is Erik Bjerneld. I'm a scientist in R&D, Uppsala, Sweden, Cytiva. And I will present the ImageQuant product line and show how it helps you to both document and analyze colonies on agar plates. Let's see. Cytiva has a long history of making scientific cameras, and this is the top of the line imager, the ImageQuant 800. It is a lab workhorse capable of many different types of imaging, and it can be configured based on user needs. Thus, it is an ideal companion if there are multiple users in a lab. It supports many applications. For example, gel electrophoresis, if you have a Comassi stain gel. It does chemiluminescence if you have a Western blot membrane. And also bioluminescence, and that is a common detection technique to measure ATP if you want to detect viable microorganisms. If you're interested in that, please just search for ATP and ImageQuant 800. We just published an application note on that topic. It can also be used to image ELISA plates, but the topic of today is to capture images of agar growth plates. So let's see. Before we look at the different types of samples, so let's take a closer look inside the camera. So at the top, you see the epi-illumination lights. They are LED lights. And depending on the configuration of the imager, different light sources and different filters are available. The epi-LED lights, they are used for color images. And they are positioned above the sample on the same side as the camera sensor. And for chemi and bioluminescence, there is no light source and no filter needed. In that case, you just collect the light that is emitted from the sample. So that is the most basic configuration. But if you want, you can also add trans illumination. That is the white light panel at the bottom of the imager. And that is used to detect the optical density, to measure how much light is absorbed by the sample, that is how much is blocked. And that can be used to quantify how much of a molecule you have in your, or how dense your colony is on the agar plate. So that is the next step, to add these white lights at the bottom. And if you are interested and have a need for it, you can also add a number of different LED lights and emission filters. You can see in the insert the default lights and filters that comes with the imager. So you see that they go all the way from UV up to near infrared. So the range, the options that are available, the different light LED combinations, they really span over the entire visible spectrum. Let's now have a look at different samples. So this is just 3 examples showing you how it can look like. To the left is a color image of a cultured membrane. For example, it could be one that Patty just described. In the middle, you see an image of an optical density grayscale image. The different colonies, they all appear black, but you can actually measure exactly how gray the different colonies are. So it is a very quantitative method if you want to measure how dense the colonies are. And then finally to the right, you see a multicolor image. And each LED light filter combination, so this is actually a composite image that is composed of many captures of this plate. Each capture has a unique light filter combination. And then you combine, you merge all these images into one, and you can select different colors for different images that was used to merge into this final multi-channel image. And as you see, different colonies light up, and that is because the autofluorescence of different colonies are different. Each type of colony has a unique, you can say, almost call it the fingerprint, an optical signature, so that different colonies light up with different light filter combinations. And this can be quite useful if you want to differentiate different colonies. And it can also be useful if you have very close colonies. You see, in this case, this was a contact sample measurement from a floor. And it was obviously not a clean floor. You see, there's a lot of microorganisms growing on this plate. But sometimes you want to distinguish, perhaps if they're growing on top of each other or maybe be very close, then this can be a very useful technique to do that. But I think for routine analysis for QC, you would not need such advanced technology. Probably you just need to analyze the color image to the left. Okay, let's continue. Now, considering bioburden testing at different stages of a biomanufacturing process, there are many types of microbiological tests which are routinely performed, just as Patty described. And to know the level of bioburden requires detecting and measuring these viable microorganisms. And colony counting is often the method of choice. It is simple and it is also reproducible. So after validating a standard operating procedure, different limits of the colony number are established, typically based on risk assessment and also baseline measurements, so that you do many measurements over time to establish a baseline, and that data is then used to set the specification, the limits for the test. And the sampling can be performed at various stages, from checking surfaces by contacting the agar plate with lab benches, floors, equipment, or clothes. You can also test solutions, just as Patty explained, using the membrane filtration technique. And you can also test the quality of air. So there is a special types of equipment for that, and then a defined volume of air is allowed to pass the surface of the agar plate before culturing the plate. So these are all standard techniques, standard types of tests. In addition, there are also tests where you actually mix the agar with the product. It could be some media, for example. Then you can mix it before casting and then see if something grows on that plate. So these are just examples of different types of tests. Now, if you test something which is pure or something which is sterile or clean, then the number of colonies can be very few. And then, of course, it might be easiest to just count them by eye. That is probably the fastest option. But there are a lot of cases where it can be convenient to actually use a software to help you. And that is when you have many plates, many colonies. They might have different shapes. They might be big and small ones in the same plate. They might have different colors. Maybe you want to distinguish different colors. So in those cases, it could actually be useful to have a software to help you in the analysis. And for this purpose, there is a software that Cytiva offers that is called ImageQuant TL. This is also a multi-purpose analysis software. It can be used to analyze gels and blot, but there is a dedicated module for colony counting. And this colony counting module, it has many nice features that I think are useful. For example, you can do automatic detection of colonies. You just highlight an area and then the software detects the colonies for you. The results are nicely presented in a table that you can export into Excel. You can create a PDF report. you can also remove artifacts and do some manual editing of the colonies. And if you want to present your data, it is possible to export high-resolution images, for example, in 3D view that you can see here in this slide. And another good feature of this software is that it's compatible with many file formats. So not only, well, the most common is .tiff and .jpeg images. So it is not only limited to our output files from our imagers, but you can use other types of cameras as well, and you can use this software to analyze those images. And this is how the software looks like when you are using it. The purple box here is used to define the area of interest. You set some settings for detection and then just click detect. And then automatically the software detects the different colonies in your plate and counts them. And you see down to the bottom left, there is a table. And in this table, not only are the colonies listed, but you can also see measurement of circularity of the colonies, of the signal from each, how dense they are or how much fluorescent signal you get from each colony, and also the area, the size of each colony. So then it's quite easy to filter out if you may suspect you have some artifacts or some outliers which are not colonies. It could be some dust, for example. That is easy to remove those artifacts from this list. Okay, I think I move on fairly quickly here. So to summarize, the ImageQuant 800 system is an ideal scientific camera, especially if you have many applications, many people using the camera. There are different models for different needs. There is anything going from that basic color document function. You can use it as a luminescence detection if you are doing ATP assays. And then you can add on different features, the optical density feature, if you want to analyze these grayscale images. And for more detailed investigations, for example, if you have been tracking a process over time and then all of a sudden you see some more impurities or some more colonies than usual, then the fluorescence can be useful as a quick first screening tool to identify the different types of colonies. And finally, we think that this software ImageQuant TL is useful to quickly analyze images and to export results. And we know that many companies, they need to comply with regulations, and we see a trend that more and more companies need to have digital archive, store their results, and show that the results are traceable, that you can have accountability so that different users, you can track who did what, and also data integrity, that you can later on go back and show images of your plates. And for these goals, then this software is very useful. That concludes my part, Patty. I think maybe the next slide is for you too. Do you want to summarize with this?
Yes. So here, let me get it bigger on my screen here so I can see that. That you've switched over to me, right? Just making sure audio is coming through. So in our products, we offer a variety of products for laboratory filtration, not just in the microbiology, but if you have any filtration need that would be for collection and recovery, protein purification. We may have a lot of individuals here from pharmaceutical that are actually in the protein research areas or the molecular biology areas. We have products for all of those research needs. We have sample preparation products for analytical chemistry. So the syringe filters or syringeless filters for sample prep as you're injecting it into your instrumentation to check the chemical quality of your products. And we have a variety of products for instrument protection. They may be venting. So good example would be in the microbiology application, any of the vacuums that are being used, whether it's house vacuum or vacuum pumps, you want to protect those with a hydrophobic filter so you don't get fluid going into that. Another area to think about that would be in any of your cell culture areas and the aspirators that you're using. You want to protect those pieces of equipment from any aerosols being drawn in, cell culture media buffers that you're actually removing from the cell culture vessels that you're working with. And then other analysis and quantitation. It may be pH papers. It may be as simple as protecting your bench surface with bench coat. So we are really a full service supplier from glass fiber and paper products to syringe filters to microbiology. So if there's anything that we can help you with, reach out to us. And, most definitely Eric's technology really plays well into the whole analysis and streamlining the microbiology workflow.
Great. Thank you so much for that informative presentation. So before we get to the question and answer session, I would like to quickly remind our audience how to submit questions. So you can submit your questions by typing them in the Q&A box, and that can be found directly below the video player. So our first question today is, can lab manifolds be scaled up for a greater number of samples?
Yes, so with laboratory manifolds, that's a good question. I only touched on it briefly during the presentation, but a lot of times too, microbiology is handled like an assembly line. So a 3-place manifold is just too short of a manifold to process, multiple samples coming in. So you may use that coupler and expand it to make it into a 6-place manifold. And like I mentioned, we've actually even done a study by placing 3 manifolds together using the couplers to make a 9-place manifold. And we do have a technical article about that that goes about explaining how we did the testing of that to ensure that the port that's farthest away from the vacuum source still has a vacuum draw that's within the range for most of the published technical specifications for membrane filtration technique. So that's a good question. Thank you.
Great, thank you so much. So on to our next question. Does colony counting with the ImageQuant system meet regulations?
Yes, yes. The short answer to that is yes. It helps meet regulatory requirements. But remember that any process needs to be validated at the user site. And also a standard operating procedure must cover the entire workflow. So the imaging and the analysis, that is just part of the workflow, but the standard operating procedure should cover also anything from the sampling to the final analysis. And just a comment, and that is that usually you talk about 3 objectives, and that is traceability, accountability, and data integrity. And the software really helps to meet these objectives. And there is even a special GXP extension. So if there are regulatory requirements for your company, then you can consider to also buy these extra software extensions. And they have extra features such as audit trails, special user roles, and also a more secure Windows login. So there are additional features for such GXP products.
Great, thank you. And is there a white paper on the ImageQuant TL analysis software?
It's not a white paper, but we have a data file that is on the website. So I would recommend to go to the website and then check out the data file first.
Great, thank you. And then this next question is in reference to the disposable funnel. What is the filtration area we consider for colony counting?
So yeah, that's an important feature of any filter funnel that you're working with, whether it's hardware or a disposable filter funnel. You really have to look at the specifications of the manufacturer. The regulations aren't going to say you have to use a certain effective filtration area. And the effective filtration area is the area that the fluid actually flows through. Usually, the effective filtration area plays a role in allowing that sample to spread out so your colonies have the chance to, spread evenly across the membrane filter and not, as they grow, get in contact or impinge upon one another. But the more colonies that end up in your sample and collected onto the membrane, the more crowded that surface area becomes. So depending on the industry that you're in, there may be ways to deal with counting of surfaces, and it has to do with knowing the effective filtration area, and that will be published. So, for example, you saw a reusable plastic funnel called a magnetic filter funnel. It has an effective filtration area of 9.6 centimeters squared. And the MicroFunnel that you also saw has 13.46 square centimeters of filtration area. So knowing what that is, and then knowing the size of your squares, if you had an overpopulated plate, just because your sample happened to be dense with organisms, you would be able to estimate what that count would be by knowing the size of the square of your grid line square, and then your effective filtration area. You could do an estimated count by counting so many vertical and so many horizontal. So, in that aspect, it certainly would be easier to have an ImageQuant image that was automatically counting an overpopulated plate like that.
Great, thank you. So moving on to our next question, how can solid samples be analyzed?
So solid samples can be analyzed if they're of a nature that can be totally dissolved in one of the various buffers, either normal saline, phosphate buffered saline, or if you're in the pharmaceutical industry, one of the various sample preparation buffers that are listed in the Bioburden chapters or the sterility testing chapters. As long as you can fully dissolve that solid in an aqueous buffer, you can analyze it by membrane filtration technique.
Perfect, thank you. And do you offer on-site demos?
Eric, do you want to start?
I'm thinking out loud here. I think so. I think that's possible, but I'm not sure. We need to maybe check that before I promise anything.
Yeah, and for the manifold and the microfunnels, yes, we do offer on-site demonstrations. We would just need to get your name to your local sales rep. And if you haven't seen the manifold in person, you really may want to schedule a demo of it. It really is a nicely designed manifold to manage any sort of contamination that you may have been experiencing with traditional manifolds.
Perfect, thank you. And is the ImageQuant 800 system able to count plates with a high total content of microorganisms having different colony sizes, and especially very small in the case of purified water samples?
Yes, that is possible. And you can also set up different criteria when you analyze using this software so that you can filter based on, for example, the size of the different colonies. So that is possible.
Great, thank you. So this next question is just asking for some clarification. So during product validation process, so especially vaccine product, from which stage can the sample be taken? And then also, what is the limit as per the guideline for what will be allowable amount for filtration?
So for your sample prep, you'd want to consult your you'd want to consult your local pharmacopoeial guidance. You're drawing a little bit on, some memory here, but it has to do with whether the sample's filterable. You have to actually, validate the method in your sample prep method. So there are various ways you can prepare your sample. You need to put your product through that testing. And in the pharmacopoeial guidance, there are tables that help you select the right sample size that you are analyzing. Eric, do you have any more experience on that?
No, I think it varies a lot depending on the process. So it is hard to give a specific number.
Yeah, and also, you have other steps that you have to do to clean up your vaccine sample of, other particulate, if it's virus and cell debris, you have to have that cleaned up. So, and then I would refer to the pharmacopoeial guidance. They do give guidance on, depending on your batch size and the nature of the sample, they give, how it's packaged. They give you the guidance on how to select your sample size, your sample volume.
Great, thank you so much. And for this next question, in the case of yeast and mold counts, is ImageQuant 800 able to correctly count different colonies with large overlapping zones on the plate?
We haven't done that specifically, but I think especially if you use fluorescence, you have a good chance of succeeding. But this is still unexplored territory with fungi samples. We have not tested that much yet.
Thank you. And are microfunnel edges hydrophobic or hydrophilic?
The edge of the membrane or the material itself is made of a polypropylene, a very pure polypropylene material. In general, it's considered hydrophobic, so it helps with the draining of the fluid off the sides of the cylinder and down through the membrane filter. But the edges of the membrane filter are not treated in a way to make them hydrophobic. But the pinch fit of the cylinder into the base and against that membrane and supportive pad underneath actually holds it very tight, so that many times when you disassemble the cylinder from the base, your edges are still dry. So you know that the fluid hasn't wicked sideways to the edges of the membrane. It's gone through the effective filtration area that was exposed by where the cylinder pinched against the membrane into the base. I think that covers both aspects, whether it was intended for the talking about the membrane itself or the sidewalls of the funnel.
Great, thank you very much. And how is membrane filtration more useful than plate pouring?
So yeah, I'll go into a little bit more detail about that. When you're using your other direct inoculation techniques, which includes pour plates, spread plates, presence-absence testing, you are limited in the sample size that you can actually place onto the top of the agar and expect it to soak in, or place into the agar and not dilute the agar so that it doesn't solidify. With membrane filtration technique, you can exceed those typical fluid volumes. And in fact, one of the typical sample sizes happens to be 100 milliliters, or even in pharmacopoeial guidance in Europe, they take 200 ml samples of their purified water. So if you look at the traditional spread plate method, typically people are using 0.1 ml. That's a tenth of an ml, and they spread it over the surface. By standard methods recommendations, you don't exceed 0.5 ml or half a milliliter that you try and spread across the surface of an agar plate. With pour plate methods, typically people are using 1 ml samples and inoculating the plate and pouring their agar in. And by standard methods in the US guidance, you don't exceed 2.5 mLs, because once you get higher than that, your agar doesn't solidify and you can't grow the colonies. With membrane filtration technique, you can process a 10 ml sample, you can process a 100 ml sample, and you concentrate it onto the membrane filter. Now, the other thing that you also can do is you can also sample small sizes. So the 0.1 ml or your 1 ml typical sizes. You just put a layer of, 10 to 15 milliliters of fluid already poured into your funnel before you inoculate your small samples. So it's very versatile from those very small samples to the larger samples up to, 100 ml, 200 ml. Some methods call for 500 ml as your typical sample size. And that's just something you can't do when you're doing a direct inoculation technique like spread plate and pour plate.
Thank you. And for this next question, you may have already touched on it, but is there a sample preparation preference in pharma QC?
So in the chapters, and I'll reference the US Pharmacopoeia, in chapters 61, 62, and 71, and when it starts talking about the sample techniques, they actually lead with a sentence that says, prepare your sample by membrane filtration technique if the nature of the sample permits. So, it's been pretty much recognized that if you can use the benefits of membrane filtration technique, start your sample prep with that, because you can separate your organisms and use various rinse buffers to remove any of the active pharmaceutical ingredient, the preservatives that might be in there, and have your best chance of encouraging the organisms, if they were in your sample, to grow. And then you validate that sample preparation technique prior to using it in your SOPs.
Great, thank you. Mm-hmm. And why is colony counting important in pharma QC?
Maybe I can start. So to know the level of bioburden in a process, it requires that you detect and you measure the viable microorganisms routinely. And colony counting is often the method of choice. I think it is because it is simple and it is reproducible. Again, you need to establish a standard operating procedure. You need to establish the limits for your tests based on risk assessment and baseline measurements. And colony counting is then used to measure the level of microbiological contamination in a process. But it could also be in a product, in a material, or in a workplace, and you measure that routinely over time. Yeah.
Perfect, thank you. And it looks like we have time for one more question before we wrap up. So to end it, why are you only using a 0.45 micromembrane in water and boarding testing?
So for most of the microbiology methods that are out there, the most referenced type of filter media is a 0.45 mixed cellulose ester that just happens to be the pore size that is called out. It gives you a nice fluid flow rate for sample preparation, which relates to your sample filtration time and how many you can process. And it has been shown to have the optimal recovery of organisms, and then still be able to draw the fluid up, or when you're the nutrients up through the membrane filter to grow the colonies at the surface. With that all said, there are some individuals that are working in companies that have water systems where they know that the nutrient content of that water system obviously is very low. This can stunt the growth, and they're worried about small stunted organisms in their water system making it through a 0.45 membrane. So they will use a 0.2 micron for their water system monitoring. And we do offer that. We do offer a 0.2 in our product portfolio. Now, if you also look at the pharmacopoeial methods, they usually state 0.45 or smaller. When you get over into food and beverage, where you might be regularly looking for yeast and mold, which are significantly larger in size to bacteria, then, they start using 0.8 micron and sometimes even 1.2 micron filters for microbial analysis.
Wonderful, thank you. And with that, it does look like it's time to wrap up. So thank you again, both for the presentation and for taking the time to answer those questions. I would also like to thank the audience for attending and participating in today's event. I'd also like to thank our sponsor, Cytiva, for making today's educational webcast possible. We would like to ask everyone in the audience to participate in a brief survey, and that will appear on your screen after today's presentation has ended. You will also receive an email alerting you when this webcast is available for replay, and we invite you to forward that announcement to your colleagues who may have missed today's live event. We hope to see you all next time. Thank you and take care.
Thank you.